Welcome, everybody, to our webinar, Recruiting Participants for Genetic Studies, What Study Teams Need to Know. So I know that this was always, personally a rather difficult topic for me as a research nurse. Whenever we had a genetic sub-study to one of our clinical trials, I always felt a little uncomfortable. I was always fearful that I was going to get questions I didn't know how to answer. And I know that I'm not alone kind of in that insecurity and kind of sort of a lack of confidence about having these discussions with our participants. So, we're really excited today to offer this webinar, and we have two fantastic speakers who are really expert in this area. Um, so you'll see they're highly qualified to lead this discussion. Both are from the NJX, which stands for Genetically Informed Research, Education, and Treatment. And they will talk a little bit more about the Great Core toward the end of their presentation. But first, Dr. Gary Heiman is a vice chair and professor in the Department of Genetics. He's also the director for the undergraduate program. His research focus is neurological and psychiatric disorders, particularly Tourette's disorder. And then Dr. Jessica Salvatore is associate professor in the Department of Psychiatry at Robert Wood Johnson Medical School, and also the director of Genes, Environment, and Neurodevelopment in Addictions research program. And Dr. Salvatore's research focus is on the genetic basis of alcohol abuse and alcoholism. So with that, we'll turn it over to our speakers. And Justin, did you have any housekeeping items?
No, I'll put it in the chat as well, but just a reminder that we're going to be doing Q&A at the end. So if you scroll down or use your mouse to go to the bottom of your Zoom app, you should see a Q&A button that's next to the green share button. And in there, you can post questions. You can upvote other questions if someone already asked one that you had in mind. You can also comment as well. And like I said, we'll address those at the end.
Okay, so we'll turn it over to you, Dr. Hyman.
No, I'm going to turn it over to Jessica.
Ah,
Well, let me just first say this is our outline for today. So, Genetics 101, a little brief overview, how to do pedigree drawing. So, if you're doing any kind of study that involves genetics, you need to know the family history. That's where this is about. Jessica will be then talking about informed consent, in particular around the genetic studies. And then, over time, we've all had who've recruited subjects for genetic studies. We get all kinds of questions, and so we have here the list of questions that we've gotten over time, and maybe some answers. But we're not gonna go through all of them very carefully and in detail. We don't have the time for that, but we will give out these slides afterwards. And then lastly, Jessica will talk about the Great Core. Okay, Jessica, going from Genetics 101.
Thank you so much, Gary, and very pleased to be here this afternoon with the group. So we wanted to start with a brief overview about genetics and really kind of give a bird's eye view of why they're important. And so, the first distinction that we wanted to make for the group is the difference between genetics and genomics. So genetics is the study of individual genes, whereas genomics is the study of the entire genome, or all of the organism or person's genes, interactions among those genes, and then the environment's role in affecting them. Do you want to advance, Gary? And so naturally the question that has brought probably many of you to this session today is, why should we study genetics and genomics as part of clinical and health related research? So we know that basically across sort of dimensions of human health and well-being, that genetic makeup or genetic predispositions are associated with risk for health and disease. And so naturally, the genetic predispositions or makeup of an individual is part and parcel of what contributes to whether someone becomes ill, and also as well as variation in whether one remains healthy or can sort of rebound following an illness. And genetic makeup can also make us more or less responsive to medical treatments. And so this gets at the issue for clinical research and the question about how do we treat patients? And really that's a cornerstone of precision medicine initiatives, because you can think of nothing more personal than the genetic variation that underlies us. And so you all may be familiar, this is a pretty well-known phenomenon, that individuals with red hair oftentimes need increased levels of anesthetics in a sort of, a surgical procedure setting. And this has to do with variation in the MC1R gene that contributes to pain tolerance. And so, these are, modest variations between each of us, but that can have really profound impacts in a medical setting. All right, Gary? And so, just to give the group a broad overview, there are many different ways to study genetics and genomics. And two of the sort of big branches that are the big distinction that I'll make for the group are between latent or inferred genetic designs and then molecular genetic designs. So on the left here, where we have the latent or inferred genetic designs, we're using information about patterns of resemblance between people of known sort of genetic similarity and difference. So when you think about twins, for example, there are two types of twins, monozygotic twins who share all of their genetic variation identical by descent, and then dizygotic twins who share roughly half of their genetic variation identical by descent. And so you can use this information about relatedness and how similar phenotypically those individuals are to make inferences about the degree to which genes influence whatever trait or outcome you're interested in. And you can use a similar logic to link, adoptees with their adoptive parent or with their biological parents to make inferences about genetic sharing across generations, and then family history, which Gary will get to in a minute here with his exercise. And then the second sort of broad study design is molecular genetic studies. And so these include things like genome-wide genotyping and sequencing, where you're actually getting information about molecular genetic variation across the genome, and being able to connect that with differences between individuals and their individual genetic makeup. All right, Gary.
Okay, then that's it for the Genetics 101 lecture. We didn't want to get too much because you could take a class and all about genetics. So now, as I mentioned before, when you're doing any kind of genetic study, you need to know if it might be if the disorder of interest might be inherited, or it's a de novo or a new mutation in that one individual. So you need to do a little family history often to look at if it might be running in the family or other disorders that might be running in the family. And so we thought it would be useful to learn a little bit about how to draw a family tree. So that's what this little exercise is about. So I'm going to go over pedigree nomenclature and help you understand the family relationships and how to ask the right questions so that you can draw the family tree. So first of all, this is the typical nomenclature for pedigrees. Squares are males, females are circles, and then if you don't know the sex of the individual, you can go that regard. Typically on a family tree where you have somebody who's affected with whatever disorder you're interested in, you sort of fill them in. And you have this person we identify as the proband. They're the people who bring the family to the clinic or to the researchers. There's other technical definitions of probands, but that's really for this purpose. Who is the affected person who brought the family to the researchers or clinicians? To identify who've been deceased, it's sort of, you put this little line across and it's sort of the fraction, not the other way around. Depending on the type of studies, you might have a lot of stillbirths or adopted into the family or adopted out of the family. I usually don't do with these symbols, I just write it underneath. The union, we don't really care as much about marriage because you can have a child without being married, is you put the line right between them, those two people. Divorce is a little line in here, although I typically like putting a double line here because it's what I learned. Consanguinity, which is, consanguine within blood, meaning that they're related more by blood. So you put a double line for when they're having union. Again, as Jessica was saying, we have monozygotic and dizygotic twins. This, nomenclature symbols puts the line here, which I don't really like because if you have spouses in there, that goes right over the person. So I have typically put it up here to indicate a monozygotic twin. And then for the dizygotic, you don't put those. And then typically with a family tree, you have the Roman numerals on the way on the left and you go across. And it's a way of finding people quickly. So, the proband here is 2-2, Roman numeral 2-2. If you have 100 people in the family and you just sort of number them willy-nilly, you're not going to be able to, take a while as you go through and try to find person 40. It's much easier to go by the generation levels and straight across. Okay? And if you're drawing a family tree and you don't remember if you asked if they had children and they say no, I usually put something like this to indicate for me because, a month later, I'm not going to remember if I asked them the question or not. And so therefore, I do that just to help understand that they have that I asked that question. And then when you have multiple unions, you're doing things like this, but I will give you a little bit more information on that. So how do you draw these multiple spouses? So, so you have this woman here who has two spouses, and her first child was with this guy right here, who passed away. And you can see how I've drawn them. And this is the second child with the second spouse. Okay, so going from oldest to youngest here. So this is the general way you're going to try to do it. They typically try to put the males first in humans, and if you're doing model organisms, it's the other way around. You put the female first. But a lot of times when you're drawing a family tree, when you have too many people, it was too many spouses, you can't always fit them in. And I just wanted to show you one family tree that I had to draw once. And just to show you how you can still do it. So this was one of the family trees that I drew, and I was surprised because you can see there are 6 children, and this is going from oldest to youngest, and the woman here, and all 6 different guys. Okay, so this is how a way of showing that you can draw all those symbols in, going from youngest to oldest and getting all those spouses in. Okay, so here's a typical sort of large family tree where to identify who all these different people are. Now just just to make it a little easier, I put the Roman numerals right next to their names rather than on the side here, just so it's a little easier because we're going to be showing different people and their different relationships. But it's really hard in this example to sort of understand the relationships. So what I've done is removed all spouses and drew a new family tree so you'll see how how people are related. And I just want to notice what I usually use is a piece of paper where I put these dashed lines across so that you can figure out who's on the same generation. When it gets pretty complicated, you want to know who's on the correct generation. I'll show you that in a couple of minutes why that's important. So this is here these two are, so let me just go this way, spouse, right? And then these are siblings. First cousins are the children of siblings, and second cousins are the children of great. So first cousins, their siblings, their parents are siblings. Second cousins, their grandparents are siblings. So this person's parent, who's grandparent, and their siblings. So this is one way of just showing that to make it a little easier. Now, often people ask, what is a first cousin once removed? And that's what this is. So we have the aunt, niece, uncle, niece relationship here. And a first cousin once removed is sort of a half step where you go from a first cousin, just to show you, here's a first cousin once removed, and a second cousin. And the reason there are different words for these is how closely related genetically a person might be. So a first cousin once removed is genetically in between a first cousin and a second cousin. And how much DNA on average they share in common. So this is important to sort of figure out as we go through some of that larger family tree, which we'll do, that you will see this information that way. Okay, so back to that family tree. So how do we, just to go through some of these, so let's just say how are these two people related? Well, you can see that they're children who's just of spouses. So they are siblings, full siblings. The next one here, we have the parents are siblings. So these two people here, this person, 2-5 and 2-2, are siblings. So these are children of siblings. So they're first cousins. And this is sort of a trick question I usually ask people. So I was going from this one to this one, and you can notice that there's there's some half siblings here, but still these two peoples are related as first cousins. Here we have the aunt and niece, so this person's mother's sister. And as I was saying before, now we have a woman who had two spouses, or really three spouses, but these two people are half siblings. And we'll be giving you the slides that you can go back and look at them closer. And we don't have the time to go through it in detail here. So here we have second cousins because their grandparents, this 2-2 and 2-5 are siblings, and the other grandparents are siblings. So those are second cousins. And here's our first cousin once removed, where we have these two, 2-5 and, 3-2 and 3-5 are first cousins, and 3-2's child to 3-5 is a first cousin once removed, as I showed before. Now, I could tell you that most people think, when I ask them, what is a first cousin once removed? They usually think it's this relationship. And this is really just the spouse of their first cousin. They're married in. That's not what once removed means, but typically most people I've talked to have said they thought the once removed meant the married in person. But I would call this the spouse of my first cousin. And here we had half siblings here for 3-5 and 3-6. And so therefore 4-4 and 4-5 are half first cousins, not complete first cousins. And going up here, we have going from this person's mom, her grandma, her grandma's sister. So that's a great aunt, great niece relationship. This is one of those ones that's sort of the language we use, but we have to identify if we're working in genetics a little different. So before I was talking about these two, but this guy over here is married in. Most people would call that person a great uncle, but if you're thinking in genetics, they're the married in. They're a person. But most people, the terms we use is a great uncle. So now, after all those family trees stuff, I do want to go over how you sort of go through the system. First, you typically do one side of the family first. Let's say the dad's side of the family. And then once you finish the dad's side, then you go to the mom's side. It's important don't let the person you're talking to take control by saying, oh, there's my first cousin here and my first cousin there, and you have no idea where they are, and it's hard to drawing your family tree becomes very disorganized. So it's if you need to take control by asking the questions. How many siblings does your father have? Are they all from the same two parents? That kind of stuff. When asking about each person, you can then ask them about their health or whatever you're trying to find out. The person, you can find out if they're alive, but if you're interested in a specific disorder or cancer or whatever, you would ask them about that. And then after drawing the whole family tree, after doing dad's side, then mom's side, then another way to do this is to ask an overall question. You can say, you told me that your first cousin John and your first cousin once removed Jane have this disorder. Is there anybody else in the family that we didn't talk about that you might remember? It's just another way for people to remember this information. First is when you're individually talking about that person, and then also as a full gestalt of the whole family. And that sometimes bring in information that they forgot to bring in the first time. So um, sometimes people, even though you're trying to take control, they'll say, oh yeah, my first cousin X, whatever that person's name, John, or my cousin X, let's be better. And you don't know how they're related. Even though they're saying they're first cousins, first of all, they may not be the first cousin. And second, they could be the married in person, or they could be a first cousin that's removed. So you don't assume that that person is a first cousin. You therefore instead you say, so let me make sure how that person's related to you. Is this person your mom's brother's child? And then you can then go through it. Okay. It's also important to note that certain cultures have different terms as you get distant. So, for example, I've heard people use auntie for any older woman in the family. That doesn't tell you very much. They may not be an aunt per se, they may be a cousin, but just because they're an older woman, that's the cultural term. So you have to be careful of the cultural terms. I try to stay away from the terms from when I'm drawing a family tree because people often get them wrong and it can be really confusing when you're trying to draw the family tree. So the standard questions you go are is how many siblings, either how many children or how many siblings did that person have? So for example, let's say you're doing dad's side. How many siblings does your father have? He has two. Okay, are they all from the same two parents? If the answer is yes, you can then go on and say who's the oldest, and then who's the next person. But if the no, if the answer is no, then you have to say, are any of them from the same? So if somebody said, I have three siblings, he has three siblings, are any of them from this, are all of them from the same two parents? No. Okay. So are any of them from the same two parents? Yes. The first and the second one are from the first set of grandparents, parents, and then the second one is through the mom. Okay, so the idea here is to get the structure in your mind before you draw it. If you assume that they're all from the same two parents, and then you find out later on, you're going to be doing a lot of racing and crossing out while you're drawing the family tree. So if you could sort of get structure in your mind, and then you can draw it and ask then questions about it, it makes it a lot easier. Issues that might come up when drawing a family tree. This happens, I've had this happen a few times. Well, so how many brothers and sisters your mom have? She has one. Okay, what was his name? Well, it used to be John, but now it's Jane. So you have to think, oh, what symbol do you use? So, and then there is some new information on how to do the symbols, and I'll show you an article about that. But typically, if you're really focusing in on the genetics, you want to know what their chromosomes are going to look like. Because when you send it, if you're doing a genetic study, and then you send it to the lab, and you say the person is female, and it turns out to be male, they're going to come back to you and say, wait a minute, you made a mistake here. So what I typically do is I'll put a square and then I'll put down, now called Jane, either had a sex change or whatever the situation is, so that you are at least getting the family tree correct when you're entering that information to a database. And then of course you want to have that information that they change because you don't want to keep making the mistake if you keep talking to this family. So it's useful to know that information, but make sure that the genetics or the chromosomes are correct. Now, that's not always easy because we have people who are XY females, and you don't know at that point, but that you'll find that out later on from the laboratory end. Second one, if you say, oh, how many, does your sibling have any children? Yes, has one daughter. She's 15 years old. Do you ask if that daughter has children? It is biologically possible, so the answer is yes. You ask that question. You can do it in a sort of a joking way. I have to ask because it is possible. Does that daughter's Jane have children yet? No, fine. People typically understand that that's not that since it's possible, you're just being careful. So other things that can happen when you're drawing a family tree. Sometimes you're drawing a family tree like those dashed lines there that I showed you before, but you have maybe 10 aunts and uncles, and therefore you're going to have a large family tree. They're not going to all fit in that piece of paper, and you can't get down to teeny little things or go off on angles. So I just typically take the next blank page, put it next to it, and keep drawing the line right across that shows the relationship, and then number the families, at the end, number the pages so that you know when they go in order. So that now you can easily just keep going ahead and you don't have to worry about like getting it smaller and smaller and smaller. I drew a family tree that was from an Amish Mennonite family. Everybody had 10 kids. So, and everybody had 10 kids. So it was a very huge pedigree, and I just kept on going as I needed to. So I'm leaving a couple of links here. These are a couple of YouTube videos about some people asking those kind of questions on drawing a family tree. And then this is an article on an updated nomenclature centered on the sex and inclusivity aspect of things. So you'll be able to click on this later on and find that article if it's something that might be relevant to the study that you're running. So that's it for my part. So going back to Jessica, we're at informed consent.
Thank you, Gary. And if you could advance.
Okay, sorry.
All right, so, part and parcel of doing a genetic study is the process of informed consent with your participants. And there's an example of a consent form from Gary's work that we're able to share with the group. And of course, the IRB is always a great resource for information, and oftentimes will direct research teams on particular language or considerations to be taken into account given the particular context and trait that you're studying in your research. And so, typically how this process unfolds, which is not going to be a surprise to this group, is really starting with a review of the consent form with the participant to ensure that they understand what is involved as part of their participation in the research study, as well as making it clear to them that they are able to opt in and opt out of whatever components offered as part of your research design. So, typically completing sort of a phenotypic battery will be separated from their participation in the genotypic data collection if you're asking for a saliva or a blood sample, for example, and making sure that they understand that they're able to make whatever choice is best for them or their family in the case of if you're asking that for a child to participate in a study. And as a process, we want to make sure that participants understand everything that's in the consent form, and also recognizing that every study is going to be different. So, the studies that are run in Gary's lab are likely different from the studies, the data collection that I work with, for example, or other groups work with. And of course, the critical component of this is for participants is knowing where their data are going, which oftentimes is dictated to some degree by who the funder of the study is. So if you want to advance to the next slide, Gary. Thank you. So I've just taken a couple of snippets from the consent form that Gary will be sharing, or from Gary's group that will be shared with this group. as sort of a resource for you all to take a look at. And the consent form gives the scientific purpose for the collection of genetic material. And it also lays out how those genetic material will be used, and importantly, what things it will not be used for. So as you'll see here in this first snippet that's closer on the left side of the page, not looking for paternity, maternity, or parentage basically of a child. They are not to be used in a sort of clinical setting, again, depending on the context of the study. And for Gary's research, these results would not be returned to the participant or to their clinicians. But, in the case of genetic incidental findings, or there being some, marker for disease that would be clinically actionable, there might be a reason for the research team to get back in touch with the participant. And again, this is all part of that really negotiated process for informed consent. And then on the lower panel here is a snippet from the consent form that lays out, what's going to happen to my data after I participate in this study. And as many of you know, federal funders in particular are, data sharing and sort of making research materials that are collected with federal funds available to the research world more broadly, as opposed to being housed in one single investigator's laboratory for the duration of their career. And so, there's oftentimes a standard language about how samples will be deposited to some of these national repositories, of which, Rutgers is a major site for national repositories of, genetic data. And as you'll see in this text here and in the sample that will be shared with the group, there are things that, as the study team collecting a particular sample, that we can control. But once these data are shared to outside resources, there actually is a sort of, I don't want to say a loss of control, but there are things that are now outside of the sort of local investigators' control in terms of data sharing, and they're sort of managed at these higher levels as part of these repositories. And so making sure that participants understand what those possibilities are is really important. And if you want to advance, Gary. And I want to be transparent that, genetic data collection is not, a topic that is, or I guess I should say, it can be controversial. So this is an article from the New York Times. At this point, it's about 15 years old. But there was this big case, back in the early 2000s that involved genetic data that had been collected by a research team at Arizona State University from a local indigenous population. And the data were used and analyzed in a way that challenged the tribe's own origin stories for their group. And so the science was sort of challenging the group's understanding of its origins and etiology, which was not what the group had consented to when they allowed researchers to come into their population. And so, this is all part of that process, making sure that participants understand how their data will be used by that local team, but as well as any, subsequent data sharing, again, where this is sort of, a bit of, the loss of local control of those data does pose an issue that you want participants to at least understand what those potential risks and benefits are. Then I think, Gary, we transition back to you.
Okay, yeah. So as I said before, we've each, I talked to a number of people like Jessica and many others who have recruited subjects for genetic studies. We've all gotten difficult questions. So I wanted to relate some of these to you. I'm not going to go over them in details because there's some different languages and stuff like different, paragraphs here. But I just wanted to just tell you sort of give you prepare you for those kind of questions. So these are the questions that you can often get during recruitment. So, will I receive the results of the genetic testing? Now, in my study, we don't consider this as testing because testing means results. And we don't have a genetic test for, Tourette's. We don't have that available. We want to create that test. So it really depends on the study. And you need to discuss this with your PI, and it should be in the consent form if there will be returning results, as Jessica was saying. If they're not returning results, there's a couple of answers that I've come up with that, or other people have come up with to answer this question. If for example, the last one, we hope that the study will provide individuals with the options of getting tested. If so, they can each decide to get tested or not. Okay, so I always tell them this. Remember, this is not a test. It means results if you're doing a test. Will I find out if I have a gene or if I have the gene or which side of the family it comes from? Again, this is very similar to the last question, and therefore I would say look at that slide before the return of results. For example, you might say, currently this disorder is only diagnosed clinically. There's no genetic test for it. We hope our research may help develop a test in the future. And not everybody would be interested in being tested, but people can then, therefore, at that point, get tested. Another controversial aspect is that typically a research study is not using a lab that have strict CLIA certified protocols. And therefore, you may be using a research lab. And therefore, you can't return results based on a research lab's protocols. They need to go through a patient testing protocols that's a CLIA certified lab. So it's something you can talk with that. And you say you could, if you want to get tested, not texted, I can refer you to a genetic counselor. How will this researcher help me? So this is a paragraph from that consent form. Basically, it's not meant to benefit them in any way. Instead, we hope that by discovering information, new treatments will be available. Maybe people can get tested, things like that, if they want. Will the samples be used for any other disorder? This is a common question, because people have heard that, I didn't realize my samples will be used for, let's say, schizophrenia, even though there's not, I volunteered on a study on, let's say, on depression. So you need to discuss this with the PI and review that consent form. It should be in there. It should say what other studies that this might be part of. Is it part of a sharing repository? In our study, it is part of a sharing repository, but it's only been identified to only use it for the disorder of interest or in associated conditions, neurodevelopmental associated conditions. So these are some answers you will be able to use from that kind of concept. Do I or my child have to give blood sample or is saliva sample sufficient? For most of the work I've done, we required a blood sample because we needed enough DNA to do many of the different types of studies, and NIH only allowed us to get blood samples. There's sometimes changes in that depending on the IRB and the NIH or whoever's funding it. Is it okay to do a saliva sample? So it's important to know, but if you're doing a saliva sample, you have to make sure that they get enough DNA. And I've had situations where a kid is, spitting into the tube and get just about the end, and then they drop it on the floor. So then we had to do it all over again. So it's really be careful. And it's hard to do, but it's certainly harder to get a blood sample from a little kid than a saliva sample. And you get more people to participate if you use a saliva sample. I haven't had this question, but somebody else told me that they got it. Will you clone me? Okay, first of all, we're not going to clone you. We can't do it. It's, the technology is not there. We're interested in trying to understand the cause of the disorder that might be in your family. And, there's no reason we're not interested in cloning an individual. Can you test for paternity? This is, a real concern for people because, you know, it's about 1% of the population that there's a non-paternity, something that I don't know, I think that was the last I saw the number was. So it really depends on the study again. You should talk with a PI. So, if they're going to give back information, this wouldn't be one question they're going to ask. I've had situations in like a genetic counseling where they say, there's a disorder in the father's side of the family, and they want to know if the fetus might be have that disorder. Well, it turns out the father is not the biological father. So the answer is no, but in situations there might be willing to do non-paternity return of results. If so, you have to do it very carefully because you might want to talk to the wife first, and then, or figure out how you're going to do it. But typically, at least in all the studies I've done, we have not returned the results for non-paternity. We do find them, but we don't return that results. So again, testing for paternity is the same thing. This goes along with other, this just continued. This is the paragraph from that consent form that Jessica was mentioning. How will my genetic information be protected? This is very a sensitive issue, and you need to really be careful with people on this because they're worried about losing insurance. Luckily, there is a federal law called the GINA, or Genetic Information Non-Discrimination Act, that helps people, protects people from genetic discrimination. So if some, insurance company finds out that they are, have a gene for a disorder, that potentially could be a problem. I remember before this act became law, there were times when I was looking at medical records of people, and I'm reading along the medical records, and it said, is participating in a genetic study of X. I'm like, that's my study. And therefore, it's in the medical records. The insurance company is going to be interested in that result. So it's just to be careful and tell people don't talk about it. Don't tell your doctors that you're participating in it unless there's something, or tell them not to put it in the medical record. And this goes along more information about the being protected. This is basically from the consent form, and the same with this. So it's just more made the language of the text a bit larger here. And that's it for those typical questions. Going on to Jessica.
Thank you, Gary. So to round out our session today, we wanted to make sure that the group was also aware that Gary and I, as well as other members of the NJX element that we are a part of, have information about how they can sort of keep the conversation going, and other resources that we offer for conducting genetically informed research. So if you want to advance, Gary.
Yep.
Thank you. And so the GREAT component is a new component as part of the renewal of the NJX award. And we are focused on the impact of doing genetically informed research and its sort of implications for translational research and translational science. And so our goal is to provide tailored instruction as well as more one-on-one mentoring and analytic support for translational researchers and translational scientists. And, the broader goal here is to help sort of people bridge the gap between knowing that genetics are important and actually being able to carry out research that is genetically informed and that is inclusive of multiple populations. You can advance. And so one of the services that we offer is a consultation service to formulate genetically informed research designs, as well as addressing issues related to sort of the ethical and logistical challenges that are inherent to conducting work in this area. And so this is available both to scientific teams and people who are sort of active in the data collection process. And then the sort of second arm of our program is sort of a community engagement component in understanding the role of genetics in human health. Next. So if you head to our, excuse me, our website on the NJAX page, you can get set up with a form to request a consultation. And the QR code, which again, these slides are going to be shared with the group for you to scan or link to later. If you press this request a consultation button, then you'll be prompted to fill in the next set of information on the next slide, Gary. Which is basically sort of a short form asking for your contact information, which consultation category you're looking for information about, and then a brief description of your problem so that we can connect you with an initial meeting to overview the consultation process and sort of take an evaluation or intake in terms of how we can help you. And then really the next steps are very individualized from there in terms of, is this for a paper someone's working on, or is this a grant that's going to be submitted? So we're happy to provide this individualized format for everyone. Next.
And that's it for that.
And so we're happy to answer any questions. I don't see any in the Q&A box, but Justin and Nancy, if you want to suggest how we handle the next part of the conversation.
Sure. So I guess if you want to put your any questions, raise your hand, I guess, and then we can unmute you to ask your question, or you could just drop it in the chat if you'd like. Well, maybe while we're waiting for folks to come forward, I have one. So I know, and maybe this is because of the patient population that I worked with for most of my career, but one of the questions that I used to get was about criminal databases. And our patients had a fear in the back of their minds that they could be linked to a crime. Do you have any words of wisdom for how to approach that concern?
Jazz, you want to do it? Or you want me to do it?
Why don't you take this question, Gary?
Okay. So, I mean, we're required by NIH and in the IRB to only do what we say we're going to do. And we would only submit that to the NIMH or NIH repositories. We do not connect them to any other databases outside of that. And the only way that they should be able to do that is if they can get NIH to give them the data, which I highly doubt NIH would give them the data. Although, I don't have any, I can't be promised that NIH, typically they don't allow for things that are outside the purview of the institute, let's say NIMH or NCI, whatever, that to be able to do that. So I would think, I certainly have no interest in it, and I'm not allowed, but because of the IRB, to do it. But when it gets to the sharing repository, I really highly doubt NIH would allow it. But Jess, do you have any thoughts on that?
Yeah, I mean, and certainly there's been recent high-profile cases where, forensic genealogy has been used to identify, for example, like the Golden State Killer and those kinds of things. Those were commercial repositories, not research repositories. But I think kind of getting back to Gary's point, whatever a authority would be able to get an order for, they would be able to do is sort of it's outside, as Gary said, the investigator's hands. But that's not to say that it couldn't be sort of forced through some other legal means. So I think it's wise that participants are asking these questions because it's part of their assessment of whether participating in this component of a study is going to be a good fit for them. I will say that in commercial repositories, probably all of us are, some fraction of our DNA is probably already represented in those commercial repositories because maybe you had a cousin, or maybe your mom or your sister or your daughter submitted a sample to 23andMe or something. And so, that's how those things work. Like they can trace the family pedigrees with the genetic pedigrees to identify people who didn't even submit a sample, which is how the Golden State Killer was caught. So
Yeah, and
Okay.
I mean, I don't even think they need the NIH data. I think they can get it from the 23andMe and these others because they're paying money. It's a clinical thing. And, as you may have heard, that 23andMe was recently sold, and that data goes with whoever's buying it. That's not how it works in a research orientation. So I think totally agree with what Jessica was saying.
Okay, thanks. So those I think are really good talking points and things that you don't always think of on the spot when you're with
Yes.
your participant. So that's appreciated. Other, I do not see any hands raised or questions in the chat. So I think you might have just about answered absolutely everybody's questions out there.
Wow, we were just thorough presentation.
Yeah, that was really very, very good. I think this group will have found that extremely helpful. And as we said, we will make the slides and the recording available, and please, you can feel free to share with anybody that you know of who wasn't able to attend today. Anything else, Justin, that I missed?
No, I think that's it. So yeah, thanks everyone for being here. And like I said, we'll be distributing everything tomorrow. So keep your eyes out for that.
And thank you, Nancy and Justin,
And thank
for
you,
inviting
Gary.
us.
Thank you, Jessica. It was a pleasure.
Yeah, thank you.
Yeah.
so much.
All right, enjoy the rest of your day, everybody.
Care.
Bye-bye.